So I guess we'll get started. Hi, everyone. Welcome and thank you for joining us for this talk. Before I get into it, I want to start by just thanking the organizers for inviting me to speak with you all about this important topic. And I also want to thank the interpreters who are joining us. So I'm here to talk to you today about whole exome sequencing. A little bit of background about me. I am a genetic counselor by training. I work at Mount Sinai, and I worked in pediatric and adult trying to find genetic diagnoses for people with various health conditions for almost 10 years. I am now the associate director of the genetic counseling program at Mount Sinai. But really, my career was very shaped by whole exome sequencing because it was a test that became available just as I was starting my career. And so I've kind of grown up in this field with this test as it's grown up as well. And so it's exciting to be able to share the advances and the changes that have happened and to talk through this with all of you. So I don't have any conflicts of interest to disclose, and my outline is very brief. But I wanted to start with some background to just get us all on the same page. So you're all here because you're interested in genetics, and I wanted to start with just thinking about what a genetic diagnosis can do for us. So traditionally, genetic testing and genetic healthcare has been primarily targeted to people who have ongoing or current health concerns. We are moving into the realm of genetic testing for people who do not have health concerns, but this is the baseline where we started. And the main reason to do genetic testing for that population has been to try to find an explanation for a genetic condition. If we have an explanation, then we can give some information about prognosis, about what could happen in the future, what is the medical management we would recommend for someone's current condition. There might be treatments. It's pretty rare that we have treatments, but there are treatments for some genetic conditions. There might be specific screening or monitoring that needs to happen, or to look for potentially developing conditions or other health concerns. But it's important to recognize that this is not a crystal ball. Most of our genetic conditions exist in a range, and so we can give people an idea of what that range is, but we won't necessarily be able to tell someone exactly how significant a health condition would be for their child if there's a range, or when a condition might start. If there's a new symptom that could come up in the future, we don't have exact information. We usually can only give a range. I also think it's important to note that an explanation can give a family some psychological well-being. Often people will wonder what the cause of something was, and may find that having an answer to that question might alleviate some potential guilt or concerns there. Having a genetic diagnosis can also allow other family members to really take control of the condition in the family. So genetics is in families, and family members share genetic information. And so if there's a genetic diagnosis in one person, other people in the family can choose to be tested, which we call cascade testing, or can choose to use that information for family planning as appropriate for them. We also have many research opportunities. There are research opportunities for people who do not have a genetic diagnosis, but there can be more specific research done and finding that research community for that diagnosis can be helpful for some families. And then finally, there are many support groups that are specific to a certain condition that can be really helpful for families as well. It can allow someone to meet other people who are facing similar challenges or having similar experiences, and find some solutions for everyday life experiences that maybe medical providers might not be as familiar with how to manage. There are many tools that we use in genetics practice for a diagnosis, and genetic testing is really just one of them. So it's really important to know the clinical picture. What is the medical concern for this person, and what are the potential genetic causes for that medical concern or multiple medical concerns? We use a tool called dysmorphology. So this is the thinking here is that if someone has a genetic change that impacts the way that their body or the way that their body works, or some health system works, then that change could also impact the way that their facial features formed or other characteristics were formed that don't have anything to do with the medical cause. They don't cause any medical problems, but they might be differences in how someone looks. And this can be really helpful. The example I like to use, we all have these sort of differences in how we look, but we really look for patterns. And so the example I like to use is Down syndrome. If you look at someone with Down syndrome, they look like their family members, but they also have similar facial features to each other. And geneticists have really outlined what these facial features are for different people or for different disorders, and are able to recognize them. So that's another tool that we use. There's other workup that often gets done. We will look at imaging like MRIs and ultrasounds and x-rays. We look at, someone's tumor types or different types of pathology. We look at other blood work, what other providers have seen for that person as well. And then of course there's genetic testing. So again, this is sort of finishing up our background information here, but if we think of our bodies are all made of cells. Our cells they're different types, but they're all cells, and most of them carry our genetic information. And the genetic information has a couple different forms that we look at. This is the image that most people are used to, is this what we call the double helix. Essentially, a gene is an instruction for the body that has a very specific spelling of these chemicals that you see here. And if there's a change in the spelling, then the gene might not be able to do its job, and that's what can cause health concerns. Genes are packaged into chromosomes, and we have, very commonly 46 chromosomes. They come in pairs. And so having sort of the right amount of information in the chromosomes is important, as well as having the spelling of the gene being what we expect is also important. And so changes in both the amount of DNA as well as the spelling of the DNA can both be different ways that we can develop health problems. So when making a diagnosis historically, as I mentioned, we focused on that clinical history and the physical exam, including looking for that dysmorphology. And then there was some baseline testing, but genetic testing was quite expensive. And so frequently what geneticists would do would be that they would try to choose the most likely gene. And if that most likely gene was negative, then we would go back and test the next gene and the next gene. And really it was just because it was cost prohibitive. It was far too expensive to try to test more than one gene at a time. It would be, maybe $1,000, $3,000 for just one gene. This is actually what has changed most for us in terms of, I shouldn't say most, this has been one of the biggest changes in genetic technology, is that, we've advanced how we look at the different types of genetic changes. We have different technology to look at different changes, but we've also been able to make our testing cheaper and faster through advanced technology, really within the last 10 years or so. So that's what has led us to this world in which we have a lot of different types of genetic testing available. Some tests will look at how a gene is spelled, and others will look at the amount of the DNA. Do we have the sort of the right number of genes and chromosomes? And so it's important to just remember that not all genetic tests are the same, and they often complement each other, that we need both tests or multiple tests to get a full picture of what's going on. We can't really just do one test to give us all the information. I won't go into this in too much more detail. I just want to clarify that there are also other genetic tests that I don't have listed here, that for different types of genetic disorders that have slightly different ways of happening. So now let's get to the meat of the topic, is what is exome sequencing? And I don't want to go into this technology in too much detail. Most physicians would not be able to describe this process. But just as a broad overview, we're trying to sequence all of the genes that we know exist. And I'll talk a little bit about what's different from a genome to an exome in a minute. But all of the genes is a lot of information. We have something like 25 to 30,000 genes. And so it's very hard to read through all of them. What we do is we chop up the DNA into little pieces. We read all of those small pieces. It's much easier to read something small than something very large. And then there's a computer algorithm that tries to put those pieces back together and line them up. So it's kind of like putting something through a shredder and then having to tape it back together and figure out where things go. And that process is what gives us the instruction manual for a person. We get the whole booklet of all of the instructions that tell our bodies how to function. The step after that is to then start to understand that instruction manual. So we look for differences in the genes that compared to a reference sequence, compared to a sample that we have said, this is what we think is sort of the healthy person genes. And so that process is quite challenging, and I'm going to talk about that a little bit more. But I also wanted to, before I get into that piece of things, I want to just back up and say, who do we use this testing for? So exome sequencing is really not necessarily the ideal test for everybody at this point in time. The first place that we started using exome sequencing was really when someone came in and there was just an unclear diagnosis. We did our sort of regular workup and it was just really hard to think of what could the possible causes be. And so you cast a really wide net. You don't have to try and pick one or two genes. You can just test them all. We also use this testing when there's a really big range of possibilities, often where it would require 2 or 3 different tests to cover all of those possibilities. We might do one test to get everything all at once. The current likelihood to get a diagnosis, or the sort of chance of a positive result of finding an answer from an exome, is thought to be about 20 to 40%. Different people will quote different numbers, and it depends a little bit on the situation of the person, the family members involved, but that's sort of the range that we give. And one of the things that's come from exome sequencing that's interesting is that we're learning quite a bit about the genetic conditions that we know about. So in the early days of exomes, we started using this technology for patients who had already tried to find a diagnosis for a long time, and really the physicians had not been able to get clear answers. And so we had a population that was maybe more complicated cases. And actually about 6% of them had more than one diagnosis, and that was why they were hard to diagnose. And so this exome technology really worked well for that. We also have seen patients who maybe don't look as their clinical history and their clinical picture don't look as much like what we classically thought of for a genetic condition. And then they got this diagnosis. And so what that means is that what we thought of for the condition is not the whole condition, and there's a bigger range than we expected. And so we're actually expanding that range and learning more about the conditions and seeing the milder end or the more severe end that we didn't recognize in the past. And then finally, we've learned a lot, and there's been a lot of what we call gene discovery and discovering new diagnoses through this technology as well. So that's all been incredibly exciting for this whole time. When we talk about that process of understanding, once we have the sequence, once we know what that person's instruction manual looks like, we have to then pull out the variations to understand if those variations could actually cause symptoms for that person. So we all have on average 20,000 variations that are different from the reference sequence. And something that's important to also remember is that our reference sequence is imperfect. So we have the sequence that we think this is what the genes should look like if they're working properly. But we know that, for example, there's an overrepresentation of European ancestry in that. And so people who are not of European background are more likely to get variations that we're not sure about that look different, but might just be normal for them. So I'll talk a little bit about that normal variation in a second, but this, I really want to just hone in on this idea that understanding that having a genetic change doesn't necessarily mean that that causes health problems. We all have genetic changes from the reference, and trying to understand which of the genetic changes cause health concerns and which are just normal variation for that person, something that makes that person different from other people, is a really hard process. And it's something that we're still working on and we're still refining. So it's not automated. There are machines that help in the process, but there are people who have to actually look at the different changes and understand what they could cause. And so if you're talking about 20,000 different changes, that's a lot of information for someone to look at. And what becomes very useful is that clinical information. So the lab people can actually use computer technology to pull out, if I list these symptoms that this patient has, which are the genes that could potentially be related to those symptoms, and pull them out and only look at those ones. And so again, I just want to note that that's another potential area where the testing might not work, because if we don't have, it will work, we'll get an answer, but we won't necessarily get the correct answer, because if we don't have enough, if we don't have the knowledge of certain genes and their clinical information, then we wouldn't be able to pull that out from the process. And it's also another reason why this testing is not currently used for healthy individuals, because we just would not be able to make sense of all of these variations if we don't have any clinical information to compare the potential genetic changes to. So in terms of actually understanding the results, so there's different ways of thinking what types of results we can get. The first is to think of it as really basic: a positive result, a negative result, and an uncertain result. A positive result meaning we found a genetic change that seems to explain the health concerns, and we'll tell you more about it. A negative meaning we did not find a change that explains anything. And then the uncertain result is this in between, we're not really sure. When the lab is looking at genetic changes, what they're looking for is to understand, they have these 5 categories that they're trying to put these variations into. And so on one end, we have, it's clearly something that we know can cause health concerns. We call that pathogenic. And on the other end, we have this benign, there's a genetic change, but we know that this does not cause health conditions, and this is just a normal variation. The gene is likely working totally fine, even though this genetic variation exists. And then in the middle is we really just don't know. And so there's a process for the lab to figure that out. So the way I think about this is, if I'm trying to spell the word cat, the right way to spell the word cat is C-A-T in the English language. And if I, accidentally misspelled it with a K, most people would read that and still know that I probably meant cat. And they can understand that word to mean cat. And so that would be an example of a benign variation. It's a change in the spelling, but the meaning is still clear. If I spell bat, if I change the C to a B, that's a totally different word. And so that could potentially be disease causing. Or if I'm just missing a part of the gene, that could also is likely to cause disease. But sometimes we really don't know. Is a variation more like a benign spelling cat with a K, or is it more like the spelling cat with a B, and now it's actually means something totally different? And so there's a process for the lab to try to understand that. And I don't want to go into this in any detail, but just I want to note a few things here. Much of the evidence that is used by the lab to try to understand this it's important evidence, and the lab will try to assess it, but it's pretty standard for that moment in time. We look at databases and see what was this gene looking like in people who we think were healthy, and we look at databases to look at what did change, or what did this spot in this gene look like for people who had this condition? Has it ever been reported before? There are a couple of areas that doing some follow-up work after the genetic testing gets an uncertain result can help to clarify whether a genetic change is disease causing or just thought to be benign. And those two areas are what we call segregation and inheritance. So how is this gene change traveling in the family? Is it something that a healthy parent has and the child who has the health condition also has, and therefore it's probably not the cause? Is the way it's being passed down what we know about this condition and how it has to be passed down in families? And then the other thing that we can sometimes do, although more limited, is what we call functional studies. So functional studies is a way to try to test the product that the gene makes to see, is the body able to read the gene properly? Functional studies are pretty hard to come by, but it is something that we could, in theory, do to try to help us understand whether a gene change is causing diseases or not. So the other thing to think about for exome sequencing is who is getting the testing. And so sometimes the only person who we can test is the person who has the health concerns. But it can actually be considered really helpful to include other family members, even if they don't have health concerns, because we're able to see how gene changes are being passed through the family right from the get-go. And so that gives us a little bit more of that variant calling classification information. The important thing to know about this is that if we're using additional family, we often feel like we're more likely to find a clear answer rather than get a lot of uncertain answers. But we can also find out other things about the family. And so if I, for example, in a pediatric setting, if I was seeing a child and I'm sending a test for the child, the mother, and the father of that child, I could actually learn if the mother was not really the mother or the father is not really the father from this testing. So that's always something that I talk to my families about before we order this test. So I talked about having a genetic change, and either we know what that genetic change does, either it causes symptoms or it doesn't cause symptoms, or we don't know what it does and it's uncertain. And so that's what we call this variant of uncertain significance, where we know what the gene itself would do if it doesn't work for someone's health, but we don't necessarily know if this genetic change is enough to cause that gene to not work. The other kind of result that we can get, still focused on the person's health concerns, is what we call a gene of uncertain significance. So we might find a variation or a change in a gene, and we don't know that that causes symptoms, but we are suspicious that it might cause symptoms. And we also don't actually even know what that gene does in the body. We might have some suspicions based on things that have been seen in other studies, like on mice or other animals, or what we know that gene's role is in the body, but we don't know what it does if it's not working. And so we don't know what the health concerns would be. So this can be important for families because it can help, again, find that research community and find out more. But it can also be frustrating because it's an answer without necessarily really being an answer. It's a lot of, we need to wait and see and find out what we can find out with more time.
I think I'm on mute. Hang on.
So I wanted to touch on this topic of the genome versus the exome, because many people have heard of genomes and don't necessarily understand why we're not doing genome testing or why we're focusing on the exome. So I just wanted to explain that a little bit. This is something that my patients would ask me about not infrequently. And so I just wanted to go back to this image. We talked about the gene being sort of a piece of this DNA. This is kind of how we think of genes looking like. So the part that's just a line it's genetic information. It looks like this strand of DNA, but we don't actually think of that as being part of the gene or what the gene finally ends up making. Instead, there are certain parts that actually go into the product and create the product of the gene. And so this is a little bit technical, but the reason it's important to know is that the exome is only looking at the parts of the gene that will make the product that we know is information that the body uses as a gene. But there is all this genetic information that is not being used in that way. That doesn't mean it's not being used. It's often involved, it might be involved in things like, whether the gene is turned off or on in a certain cell, but it's not impacting necessarily the genes, what the gene makes. It might be impacting sort of how the gene is used. And the reality is that we're not very good at understanding that part of things. So we understand quite a bit about genes. We understand quite a bit about changes in genes. And obviously I've been explaining, there's things we don't understand, but this in between part is hard for us to understand. And that's the main reason that we're not really doing this testing clinically that frequently. It is becoming available really in the last couple of years to do what we call whole genome sequencing instead of whole exome sequencing. But right now it's not really clear how much more likely we are to find an answer by doing a genome as compared to doing an exome, just because we get a lot of more information, but we don't know how to interpret all that extra information. And it's also just important to note that, these tests take a long time. Genomes are incredibly expensive at this time. They're generally not being covered by insurance. And so all of that also plays into why we're still really focusing on exomes and happy to use exomes and feel like that's an appropriate test. So the advantages of doing an exome as compared to other testing, and I just want to back up and say that the other testing that we could do to try to look at genes in the same way as an exome is what we call a panel. So a panel is really a subset of genes that we think are important for a specific condition. So sometimes you might have a panel for, if you're looking for a reason for intellectual disability, there might be an intellectual disability panel that has, a few hundred, sometimes more than that, genes that they're looking at, as compared to looking at trying to look at all of the genes. And so the advantages compared to trying to pick a panel or a couple of panels to trying to look at an exome is that this is a really broad approach. We don't need to know what we're looking for. We can just do the test and see what it comes out and really try to then make sense of the results. It can be faster depending on how clear a diagnosis is. It can be faster than trying to pick a few genes at a time and then repeat and do other testing until we find an answer. And it can potentially be more cost effective too than trying to do multiple different tests. Not necessarily though. But it's important to know that there are things that can be missed. So, for one thing I talked about, there are many other mechanisms that can cause genetic conditions, different kinds of genetic changes that would not be seen by doing this testing. It's not looking at the genome, so we're not getting that regulatory information, but again, we're not really sure what that information is doing for the most part. And then there's a concept of coverage, and it's a hard concept to explain, but it's an important piece. Because this is the main reason why we might choose to do a panel over an exome for a specific person. So when we're looking at genes, it's a lot of information. We have 25 to 30,000 genes, and we're reading a book for each gene. And so really, when we're trying to find a diagnosis, it's a bit like looking for a needle in a haystack. We're trying to find a single letter change out of all of that information. And so the way I think about it is if you're doing a panel, you're basically saying, these are the regions, these are the small parts of the haystack that we should focus on because we're pretty sure that the needle is going to be in there. And, just like anything else, if you're looking at small parts, you can look in a much more detailed view. If you're trying to look at the whole haystack, you can't go in with as much detail. So that's really how I think about it, that it's possible to miss something on an exome because it just doesn't give as much depth in trying to read through and as much detail. And so that's, again, just an important thing to note, that your provider, if you have a clear specific diagnosis in mind, it might be better to go for a clear specific diagnosis test rather than trying to do this really broad test and find out a lot of information. Okay, so that is all the information that's, well, not all the information, but that's important information for the main reason for doing the exome. But there's a second type of result that can arise from this testing and is part of our discussions for every patient that we talk about. And that's these secondary or incidental findings. So as of last year, there are 73 genes that the sort of governing bodies of genetics have decided to should or could be reported to families that have nothing to do with the reason a person is getting an exome, but could be important information for that family in terms of managing their health. And one of the things that's really important is that all of these genes cause, if there's changes and they're not working, they cause conditions which are considered actionable. There is something that can be done to try to either prevent or mitigate the symptoms. So I break them up into these three categories. Some of them are cancer predispositions. Some of them might cause changes in the structure of the heart or the rhythm of the heart. And then some of them have a different type of, basically a different subset that are just sort of miscellaneous. And so all of these, again, there are things that could be done. Someone can get MRIs of to screen for cancer, or have a surgery to remove the tissue if it's not important tissue to prevent it. So for example, if there's an increased risk of breast cancer, someone could either choose to have more frequent, earlier mammograms, MRIs, or they could choose to remove the breast tissue and really reduce the risk of that cancer. And it's similar for cardiac or these other miscellaneous conditions. There's always something that could be done to try to prevent or catch something early. It's important to just note that this means that the lab will not report anything that they find. They will only report something that's related to the condition the person is getting the test for, or these secondary findings if people choose to get that information. They will not report something like Parkinson's if it's not related to the symptoms of the patient, because there's really no treatment or management that would be recommended at this time. It's also only reported if it is something that seems like it would cause symptoms. So if it's something that's uncertain, it would not be reported. And that's really important because it's important for families to know that if there is one of these concerns, like a cancer syndrome or a cardiac syndrome, there's a family history, for example, this is not the best test for them to do. It's much better for them to have that direct test looking for that, because it might not be reported based on this test. This test is looking for the other health condition. So again, if I'm seeing a child with a developmental delay, they're not meeting their milestones, families can choose to learn about a cancer diagnosis from this test, but it wouldn't be as good as them seeing a cancer genetic counselor and getting a cancer focused test if they wanted to really learn about that family history. It's also really important to note that this is optional. You can do the primary part of the test and choose not to get this information, or you can choose to get this information. And it's really a personal choice for each person, whether they want this information or not. And there's many reasons that some people do and many reasons that some people don't. But it's important that the different family members agree if more than one family member is being tested on what the plan is. And the reason for that is, again, if I'm testing a child and two parents, and we want to get this information for the child and we want to get this information for one parent, but not the other parent, that could create a really challenging situation where now the child is found to have something, the parent who wanted to know was not found to have that same change, but the other parent actually didn't want to know. And now we can sort of assume that it came from that parent, and now they're being forced to know without wanting to. So as much as possible, we really try to, talk this out and have everyone feel comfortable and on the same page about getting information or not getting this information. The other thing to consider when thinking about these secondary findings is genetic discrimination. And I really focus on it in terms of the secondary findings because The protections and the risks of genetic discrimination are a little bit different for people who have symptoms as compared to people who don't have any symptoms. So people who don't have any symptoms, things like their health insurance, or they would have no reason to think that they have a genetic condition. But people who have symptoms, that's already sort of known and recognized by insurance or other insurance providers. The nice thing about genetic discrimination is that in the United States, we do have the Genetic Information Non-Discrimination Act, which does protect us from some types of discrimination. This was signed by George W. Bush back in 2008. And this act protects us from discrimination by health insurance and from employers. So your health insurance is not allowed to charge you a higher premium, deny you care or coverage based on a genetic result, and employers are not allowed to hire or fire or ask someone to have a genetic test. They're really not allowed to have your genetic information unless you share it with them. There are exceptions within this. So for example, the military is an exception. The federal government, employees of the federal government are an exception. Employers with fewer than 15 employees is another exception. So it doesn't cover all of these situations. But it's also important to note that it doesn't cover disability insurance, long-term care, or life insurance. And so this is another consideration for our families. And I often talk with families who sometimes, will purchase a life insurance plan prior to doing the testing just in case something is found. So I did want to spend a little bit of time talking about costs. The most up-to-date, sort of lowest end of cost, if you're trying to get an exome and you want to just pay out of pocket, is about $800 to $1,000. $800 for just one person, $1,000 would be for 3 people. And that's one lab. Different labs have different costs. So this is really the lowest that we've seen in our practice here. Insurance coverage for this testing is really like everything else, depending on the individual insurance plan. Some insurances will cover this on a case-by-case. Some have very specific criteria that they use, and you need a genetics trained professional, like a physician, a genetic counselor, or a nurse with genetics training to fill in a form and sign to have this testing. And then some insurances I've worked with have really just chosen to not cover this testing at all. And this is an area that I think we can continue to educate our insurance providers about why this testing is important, is useful, is potentially cost effective. And my recommendations for families is just always ask if you can have a benefits investigation done. This is a procedure in which either your provider or the laboratory can actually often do this for you as well. They will contact the insurance and just get a sense of what is the likely out-of-pocket cost based on your deductible, your plan, whether it's in-network or out-of-network with your plan. And so you can get some actually pretty comprehensive information, and some of the labs will really stick with the cost according to the benefits investigation, even if later on it's not covered as much as they expected. So I think it's a good thing to do before doing the test, because it could be quite expensive otherwise. And then the last concept I want to hit on really quickly is the idea of reanalysis. So I mentioned that 20 to 40% of the exomes that we send, we expect to get a positive result and find an answer, but that still leaves 60 to 80% in which we're not getting that answer, even though we're reading all of the genes. And the reason for that is really this chart of all of these unknowns. Of those 25 to 30,000 genes, we know what maybe a third of them actually do. And our hope is that as time goes on, we continue to do research, collect more information, more people get this testing. And so if the first time around the result is not clear or not positive, the test is still useful moving forward. And so we call this reanalysis, that within a certain time frame, be it a year, a couple years, the lab can go back and just look at the data. So nobody has to have any more blood draws. The timing is much faster as well, typically, because they're just looking at the sequence that we already know and they have kept that in their database. and they can go back and read through and update the testing with what we know at this point in time. And so that can also help us moving forward, try to get that diagnosis. And it's kind of nice to know that it's not really at this point worthwhile repeating the test for the most part. It's just reviewing the information and trying to update the information and the interpretation of the test. Some labs do this for free and some might have a cost associated. So my advice for anybody who is considering doing this test is just asking if it makes sense for you. Take time to really understand, what is the team thinking when they're talking about doing this testing or a different test. If you want this test, you can certainly ask, but be open to the idea that the team might have reasons why they don't feel that it's appropriate. And then in terms of the secondary findings, just really take your time to think about what you would want, discuss it within, the people who are actually getting tested to try to come to an agreement. Genetic counselors are available to help have that conversation. So I think that is something that we see as part of our role and are happy to have that conversation and be a sounding board and try to come to a consensus. Very rarely I have families that just really find this decision very hard and have actually gone to a therapist and sort of like a couples counseling setting or a family counseling setting to really come to a decision. And if you decide to do it, think about the discrimination, considering purchasing some insurance plans beforehand, particularly that life insurance plan, and consider speaking with other family members who are not involved in the test about if they would want to learn these types of results. I think it's better to have those conversations beforehand rather than saying, oh, I found something, now do you want to know? It's a little easier for them to think about if they want the information when it's hypothetical. And I didn't want to talk too long about where this is going, but I've clearly focused on sort of the pediatric or the adult setting of people looking for diagnoses. There is more happening for the world of cancer syndromes, and actually this test is now being used much more frequently in the prenatal setting as well. So with that, I know that was quick. It was a lot to cram in, but I have a couple minutes for questions. I do see one in the chat about how far out can a reanalysis be done. It really is lab dependent, but most of the time the labs will really work with you. And I haven't seen like a latest date. I think they're happy to do it at any point after the testing is done. I personally, and I don't have great data on this, but my personal feeling is that probably at least a year should go by so that you have the best chance of actually finding new information from the reanalysis. I think if it's done too quickly, our field advances fast, but it maybe not quite that fast enough to do it sooner than a year. Any other questions I can answer? I can actually stop sharing too. I'm so happy that everyone decided to join us today. I hope this was useful. I'm going to just put my email in the chat. If anyone has questions, I'm always happy to try to answer those questions.